Hey everyone, and welcome to This Explainer. Today we're going to be exploring the hidden geography of our own bodies. We're going to map out our incredible immune defenses and look at exactly how they are strategically split into two entirely distinct systems. If you've ever wondered how your body actually trains its internal army before deploying it to fight off infections, well, you are definitely in the right place.
I mean, think about it for a second. If I were to ask you where your heart is, you can just point right to your chest. If I ask about your lungs, you know exactly where they sit. But where exactly is your immune system? Unlike most organ systems that are confined to just one specific anatomical location, your immune defenses are widely distributed all throughout your body. It's really this massive interconnected web. So to understand this web, we first need to define the structures that actually make it up, which are the lymphoid organs. And why are they called that? Simply because they contain massive numbers of lymphocytes. Those are the white blood cells that essentially act as your body's specialized soldiers. These highly organized tissues are the command centers that protect us from infectious agents.
Here's our quick roadmap for today. We're going to cover the immune network, primary organs, which are the boot camp, secondary organs, or the battlefield, a quick look at mammals versus birds, and finally, why it matters.
All right, let's jump right into Section 1, the immune system network and overview.
This right here is the absolute foundation of everything we're discussing today, the primary versus secondary distinction. Your lymphoid organs are divided into two very distinct groups. Primary organs are the sites of lymphocyte development and maturation. And the key takeaway here? There's absolutely zero antigen exposure. It is a highly controlled, totally isolated environment. Secondary organs, on the other hand, are where the actual immune response and memory formation happen. That means, yes, they are fully exposed to antigens and pathogens. It's literally the difference between a secluded training facility and the chaotic front lines of a war. Let's track a single immune cell's journey so you can see how this all connects. Step 1. Our lymphocyte is born in a primary organ, the bone marrow. Step 2. Some of these cells migrate to another primary organ, the thymus, for some really intense isolated training. Step 3. Once they're fully trained and mature, they are deployed out to a secondary organ, like a lymph node. And finally, Step 4. They just wait there until they encounter an antigen and spring into action. It is a beautifully choreographed transition from boot camp to battlefield.
Moving right along to Section 2, primary organs, the boot camp.
So in mammals like us, the primary boot camps are the bone marrow and the thymus. The bone marrow produces all your blood cells from stem cells, and specifically, it's where B cells mature. The thymus, which sits right in your chest, takes on the T cells. Inside the thymus, T cells undergo extremely rigorous positive and negative selection. This basically ensures they can recognize foreign threats without accidentally attacking your own body, a totally crucial process known as immunological tolerance. And remember, all of this boot camp training happens before these cells ever see a real pathogen.
Now here's a really fascinating detail about the unique life cycle of the thymus. It actually produces its own hormones, things like thymosin, thymopoietin, and thymulin, to carefully regulate all that T-cell development. But incredibly, it undergoes what's called physiological involution. That's just a fancy way of saying that as we age, the thymus literally shrinks and gets replaced by fat. It's largest and most active when we're young and really need to build up our naive T-cell army, and then it kind of slowly retires as we grow older.
Okay, on to Section 3, secondary organs, the battlefield.
Once those fully immunocompetent lymphocytes leave the primary boot camps, they head to these secondary outposts, scattered strategically throughout the body. And there is just an incredible diversity here. We've got lymph nodes filtering your tissue fluid, the spleen filtering your blood, and these massive networks of specialized tissues guarding all your vulnerable surfaces. We have MALT in the mucosa, GALT guarding the gut with Peyer's patches, BALT in the lungs, and of course, your tonsils acting as the first line of defense in your throat to trap inhaled microbes. These are active war zones designed specifically to trap antigens so your deployed cells can multiply and attack.
If we contrast the two major secondary organs, we see a really brilliant division of labor. The spleen is the largest lymphoid organ, and its specific job is to filter the blood itself, trapping blood-borne microorganisms and even recycling old red blood cells, or erythrocytes. The lymph nodes, on the other hand, are these tiny bean-shaped structures that act as local filters. They filter the lymph fluid draining from your tissues, which ensures any local infection is trapped and dealt with before it can spread further.
All right, Section 4 introduces a really fun twist: mammals versus birds.
It turns out birds have a highly specialized primary lymphoid organ that we mammals simply do not possess. It's called the bursa of Fabricius. It's this unique sac-like structure located right near the cloaca.
While mammals mature their B cells directly in the bone marrow, birds use this highly dedicated bursa specifically as their primary boot camp for B cell maturation and the development of their antibody-producing cells.
And honestly, I absolutely love this piece of trivia. When you hear about B cells or B lymphocytes in your own human body, you probably just assume the B stands for bone marrow, right? Well, actually, scratch that, it doesn't. The cells were first discovered in poultry, and the B actually stands for the bursa of Fabricaceus. The scientific community just kept the name even when we later discovered that mammals use bone marrow for the exact same function. How crazy is that?
To quickly summarize the structural differences here, both mammals and birds have bone marrow and a thymus. But while the bursa is completely absent in mammals, birds rely on it heavily. Conversely, while mammals have a vast, intricate network of lymph nodes, birds have very poorly developed lymph nodes. So they rely much more heavily on other secondary tissues and their bursa to manage their immune responses.
Which brings us to our final section, why this distinction matters.
So, knowing whether an organ is a primary boot camp or a secondary battlefield isn't just fun biology trivia. It is literally the foundation of clinical medicine and pathology. Doctors and veterinarians rely on this exact anatomical map every single day. If you're trying to develop an effective vaccine, you have to understand how to stimulate those secondary organs to create memory cells. If a patient has an immunodeficiency disorder, you need to know, did the primary boot camps fail to train the cells, or are the secondary battlefields failing to deploy them properly?
Certain diseases actually strategically target our immune map. For example, Gumboro disease in poultry specifically targets and destroys the bursa, a primary organ. This means it literally wipes out the entire B-cell boot camp, causing severe systemic immunosuppression because new cells just can't mature. Leukemia starts in the bone marrow, which is another primary site. On the flip side, lymphoma targets the lymph nodes, wreaking havoc in the secondary battlefields where mature cells are supposed to be actively fighting. Understanding exactly which organ type is affected tells a clinician exactly how the patient's defenses are breaking down.
Let's look at one more concrete clinical correlation regarding those secondary organs. Sometimes, due to trauma or severe disease, a patient needs a splenectomy, the surgical removal of the spleen. Now remember, the spleen is the major battlefield for filtering blood. Removing it significantly increases the patient's susceptibility to infections caused by certain encapsulated bacteria. Without that specific secondary organ trapping blood-borne pathogens, those bacteria can just multiply unchecked in the bloodstream. The body's defense network has lost a totally critical outpost.
I want to leave you with this final provocative thought. We know some of the most devastating diseases out there destroy the primary boot camps, the bone marrow, the thymus, the bursa, stopping immune defense before it can even start. So if those training grounds are compromised, how could we engineer future vaccines or therapies to totally bypass that primary damage and somehow deliver pre-trained defenses directly into the secondary battlefields? It's a massive challenge at the absolute forefront of immunology today, and finding the answer relies entirely on understanding this incredible, dual-layered map of our immune system. Thank you so much for joining me on this explainer, and keep questioning how your amazing body works.